I love French fries. No, literally, I do love them. I love them. And I think we all love them, right? And I sincerely hope that you guys love them, because to be honest, they are just so amazing. They are. And yes, okay, I know that French fries are basically just potatoes. I know that. I've been told. And yes, speaking of potatoes, do you know how many potatoes are produced in the world every year? No? No. 374 million tons. This number seems absolutely huge, right? It seems enormous. But it doesn't even include the huge percentage that is lost to pests and diseases every year. I mean, please, just take a moment and imagine how many more French fries we could have if we didn't lose so much to pests. I mean, please, just take, sorry.
Madeleine, we talked about this before. Excuse her, she's too much into French fries. It's a bit of a problem. But the truth is, she's right. 40% of the global agricultural production is lost to pests every day. And we're not just talking about potatoes here. The real problem is the impact on food security. And as you must have heard 1,000 times, climate disruption is causing a huge loss of arable land through flooding and drought. Climate change is clearly leading to the unpredictable and significant proliferation of devastating pathogens, giving rise to a huge uncertainty. We felt like it was important to exchange about this with the farmers as the people providing us with food. And the people we met expressed a clear need for a change. A change because they are struggling to adapt, to provide enough food that is good and healthy. They are dependent on their productivity, which is drastically decreasing. They are under strong pressure from authorities that are pushing them to use less pesticides, all the while being incapable of providing them with alternatives. And as if it was not enough, they are feeling the increasing pressure from food industries as customers are extremely demanding regarding food. We were literally told that constraints are coming from everywhere.
Whoa, thank you, Annelle, such a drama queen. I'm sorry about that. But you know what? Let's not be so dramatic about it. And instead, let's focus on helping them. So the question is, how can we use our knowledge in synthetic biology and engineering to address this global problem? Or in other words, how can we help scientists to help farmers? Well, to do that, we exchanged the topic with a lot of scientists and farmers, and we realized that there is a huge lack of knowledge on a huge amount of unknown pathogens. Because even if scientists really wanted to observe and understand bacteria's behavior in situ and real time, well, they can't. Because they don't have the methods that are reliable enough and efficient enough to do that. An example, here is a rotten potato. If you want to understand what happened, you can just, like on this image, cut it open and see what's inside. But that's not enough. You don't know what pathogen it is. How did it develop? At what speed? At what optimal growth condition? You know next to nothing. So what science needs is a tool that is adaptable, safe, visual, in real time, non-destructive, in order to allow in situ studies. Because if tomorrow a new pathogen emerges, and this will most certainly happen with the climate change and everything, we have to be ready for it. We have to be ready to react quickly and address this global problem.
And to do this, we had a dream. More French fries. No, I'm just kidding. Am I? Imagine, sorry, fuck, no. Okay, no, we had a dream. What if we could simply see the invisible world of bacteria by just lighting them up? And I mean, we didn't have to reinvent anything, because as always, nature is one step ahead of us. All we had to do was adapt the natural system that produces light, the Lux operon from the P. luminescens bacteria. So you may be wondering, great, bacteria lighting up, how does it work? So we have Lux A and Lux B. Now, these two genes code for the luciferase, so the enzyme that catalyzes the reaction producing light. And then we have Lux C, D, and E. Now, these three genes code for an enzyme that recycles the substrate that is needed for the reaction. So this is the Lux operon. And it was actually enhanced in 2018 into the so-called iLux opron, which is capable of a 7-fold increase in light emission, which is absolutely huge, right? It is, it's huge. Now, we built upon this system, went even further to propose one that is 100% accessible and adaptable. Basically, we have created Fiat Lux, a universal tool.
Right, yes, but I think we should say how we use this opron exactly. So let's dive into the science a bit more. I promise I'll be fast, so hang in there. What we did was built on the work of previous iGEM teams who worked on bioluminescence, such as iGlowy in 2010 here, to go even further and propose a concrete application of the system. We took our Fiat Lux opron in a biobrick format and inserted it into the conjugative plasmid PCVA, and we added a constitutive promoter that already exists in the registry. Then this plasmid can be used for conjugation to introduce it into many bacteria so that they produce light, like here. Our construction enables us to conjugate crop pathogens, and by lighting them up, we can see them to try to understand them. Okay, Laia, but that's the theory, right? Now we have to test our tool on a real-life problem. And for that, we wondered, why not testing it on potato bacteria to please Madeleine and her French fries addiction? Now, yeah, that was a joke. We exchanged with farmers about it, and they all put forward the blackleg disease of potatoes. This disease is caused by the bacterium Dicea solani, and this causes the blackening of the plant and the rotting of the tubers, making them all impossible to commercialize. So basically, blackleg is our pet peeve. This is a highlight quote that we remembered from our many encounters with potato plant producers. So this is why we chose Dicea solani as our proof of concept bacterium. Now what we did basically was take this bacterium, conjugate it with our conjugative plasmid in order to make them bioluminescent. So remember, this was easy for us to do because our plasmid is conjugative. We had our luminescent bacteria. We had to test it on plants. And for that, we chose chicory leaves because it's often used in labs. We infected these chicory leaves, and we observed the propagation of the bacterium inside the plants in real time for 24 hours, and this was successful. Yeah, so what you can see here is amazing. We can see our bacteria propagating in real time. We had our proof of concept, it works. Now the next step was to make sure it also worked in other bacterial strains. And we had time to test several of them. We had Dicea solani, of course. We also tested Citrobacter rodentium, which is a lab mice pathogen. And it worked. So this is great news because it shows that it not only works on plant pathogens, but also in animal pathogens. We also tested Pseudomonas putida, which is a bacterium present in soils of many different ecological habitats. And it also worked. So, I mean, it really shows the versatility of our tool. Can you imagine its potential?
Of course, Mathilde, but not all is great yet, because we still have an issue. How do we detect the luminescence? Well, for that, some machines already exist, sure. They cost at least $30,000. Clearly, not all labs can afford this kind of machine. We felt that it would highly limit the use of our tool. But after a few more tests, we realized that the plasmid not only works, but produces much more light than we expected. Here is a photo we took with just an iPhone camera. So these results encouraged us to look for alternative solutions, and we came up with the idea of developing a hardware device, a box that creates a dark chamber designed to efficiently capture bioluminescent images and to test different conditions by controlling the temperature and the humidity. The box is designed so that you can use a smartphone or a camera, and we developed a camera piloting software for Raspberry Pi to make the system complete and independent. To test the box, we infected potatoes with our luminescent Dicea solani, and we obtained these kind of images. The hardware costs less than $400. It is affordable, accessible, adaptive, and easy to use. So basically, it allows anyone to use Fiat Lux.
Yes, Laia, but to understand the bacteria, you also have to analyze those images. And the existing softwares for scientists are far too complicated for the simple analysis of luminescence. They are very often not open source, not customizable, don't give access to the unprocessed images. So our goal was to build a software for biologists by biologists that is open source, user-friendly, and 100% transparent. And we did it. Our software is made up of 3 different parts: data import, processing, and visualization. During the import of a time-lapse, such as the one we saw before, for example, users can subcut the image, preventing them from having to do multiple operations on the same plant, and they simply have to fill in information such as the scale and infection point. The processing step includes detection of the plant area, of the luminescence area, and luminescence clusters, and all intermediate readable data are available to the user. Thanks to these steps, users can visualize quantitatively the intensity on a graph at a given time or over time. They can also make a virtual dissection and visualize the intensity there. They can then develop mathematical models and conduct a thorough analysis. This software made a crucial difference to our project, as it allowed us to truly link the wet lab part and the dry lab part, even for non-data analysts. This was the missing piece of the puzzle, giving our tool complete autonomy.
Exactly. So to close the loop, our tool comes as a service package. The plasmid, coupled to our hardware and software, allow us to see the bacteria so that we can understand them in order to be able to fight them. Our tool fulfills all the goals we had set with all the encounters with scientists and producers. Fiat Lux is efficient, accessible, adaptable, safe, visual, in real time, non-destructive, affordable, and autonomous. It works. This is a game-changer tool for scientists that would greatly improve our current agricultural practices. This is a true creator of knowledge.
Now, one word that really stands out here is autonomy. Because by providing biologists with open source software and open source hardware, we are not only providing them with an autonomous biological system, no, we are providing them with an autonomous tool. So you may be wondering, great, how can Fiat Lux be used tomorrow, concretely? For example, to save French fries. I'm just putting it out there. So if a pathogen appears in crops tomorrow, there is a high chance of this happening, samples can simply be sent to a Fiat Lux lab, where the pathogen will be isolated and then identified. Then Fiat Lux will be used to study the pathogen. Now, what do we mean by that? We mean transferring the plasmid into the bacterial strain. We mean infecting a new and healthy plant with this bioluminescent bacterial strain. We mean observing the luminescence thanks to our hardware and analyzing the results thanks to our software. Now, all of this will be very helpful in 2 particular cases. For example, if the pathogen is completely new, completely unknown, basically we know next to nothing about this pathogen, Fiat Lux will be essential to understand it, understand its propagation, its optimal growth conditions, for example. Now, in the second case where the pathogen is known, but to this day there are no known treatments, Fiat Lux will be essential. And please just take a moment and think about it, because all of this could be done in a matter of days.
Which is so important because it allows us to skip to the next phase more efficiently in order to have more time to focus on the development of new treatments, the selection of specific varieties, or the development of new biocontrol methods. So at the end, who will benefit from our tool? Well, first, academic labs, then R&D teams in the crop science field, and in the longer term, the farmers. Because all the solutions found thanks to Fiat Lux will eventually help them to save their crops in order to produce better, waste less, with no price increase. And to note that our main sponsor, Innov3PT, which is the French Federation of Potato Plant Producers, is already interested in our tool because it will help them to test several biocontrol methods on potatoes directly.
Now, we strongly encourage future iGEM teams to build upon our work and go beyond our project. Now, these next steps could include, for example, using Fiat Lux as a reporter gene, or extending the use of Fiat Lux to many different bacterial pathogens. Future iGEM teams could also perfect the characterization of our tool, or increase the plasmid stability with a toxin-antitoxin system. And side note, we have already created the biobricks to do so. Finally, future iGEM teams could also adapt the construction to other types of pathogens and not only bacteria. Now, Fiat Lux will be essential to address the United Nations Sustainable Development Goals, which include zero hunger, responsible consumption and production, and climate action. In any case, all of the stakeholders and members of the general public that we have interacted with are convinced by Fiat Lux, and now we hope that you are too. Thank you very much. And thank you so much to our sponsors, Innov 3PT and Biomérieux. Thank you so much. To our PIs too. Thank you.